This is a little bit of an unusual talk. I have titled it Computational Microscopy of SARS-CoV-2. And I'm going to tell you about our modeling work on predominantly I'll focus on the spike protein, the SARS-CoV-2 spike protein. I'll tell you a little bit about some of the overall goals of our effort that actually sort of link to trying to model the whole viral envelope. But I'm going to start off with just sort of a very high level introduction in general about the methods and the kinds of questions that we can ask and answer and why people are interested to build these types of models and so forth before I get into the details of SARS-CoV-2. I'll also say this talk is somewhat unusual because we are really just getting started with what we're doing. You know, the data is being written to disk right now, and so we're analyzing as we go, and we just really started that so recently. So, you know, ordinarily when one gives talks, you're typically talking about sort of data that you've really, you know, thought through carefully already, but this is a little bit by the seat of our pants. But in any case, so thanks for having me. So let's get started. Okay, so my group has been one that has been very excited and sort of always enthusiastic about trying to harness the ever increasing, really tremendous growth of compute power that we've had available to solve outstanding questions in sort of complex biological systems. We've had an emphasis for many years on trying to use this data to develop new therapeutics, in particular where the dynamical motions of molecules that can't be seen with experiment can somehow be probed and explored and predicted with computational methods that I'll talk about, mainly all-atom molecular dynamics simulations, and then used in the discovery and design of novel therapeutics. And so I'm sure, like this crew in particular that I'm talking to, you certainly appreciate most likely that the real advances in science are going to come at the intersection or the convergence of high performance computing, data science, and enormous, you know, incredible data sources. That's where we're going to really make the big advances. And so again, I probably don't need to go over this. This is just to indicate, you know, that we've had this great rise in compute power every 4 to 5 years, going up by an order of magnitude. You know, we're among the users of these petascale machines in the United States, and the, you know, sort of always looking forward to the next machine. And basically what this has allowed us to do over time has been to increase the complexity of the systems that we are interested to model. So, you know, originally people started basically with systems that were on the order of a single protein with about 10,000 atoms and sampling timescales on the order of hundreds of picoseconds. And then, you know, with this increasing power of architectures, we've been able to increase the complexity of the systems, going to membrane-bound ion channels, sort of large molecular machines like ATPase, the ribosome. I'll tell you about our work with Influenza, which is this little rotating ball that you see there on the right of the screen. And then in addition to the complexity of the systems and the size of the systems, of course, the timescales that we're able to reach also have been correspondingly increasing. And for questions in biology, it's very important for us to be able to get out to, you know, milliseconds, even seconds or longer, you know, so those are where a lot of the interesting questions are. So there's lots of folks trying to develop, you know, creative ways algorithmically to, and methodologically to sort of access these longer timescales. Okay, and so, yeah, again, sort of reiterating what I sort of said is that the real research breakthroughs, you know, we think are going to occur at the interface of experimental observational science and simulation science, and be the methods and the approaches that are going to allow us to address, you know, the biggest changes, challenges facing humanity, things like climate change, for example, developing sustainable energy materials, and then also the development of new therapeutics, which is where I'm going to focus, of course, the talk today, and trying to sort of unite these approaches to get new insights particular to SARS-CoV-2. Right. So the main sort of a very nice analogy, I think, sort of an intuitive analogy of these methods is that is to think of them as a so-called computational microscope. And the idea here is that we are starting with experimental data and then using these numerical algorithms to basically explore the time-dependent dynamics of our system and then get some new insight that really takes us, sometimes it takes us, oftentimes it takes us beyond where experiments can go. It helps us to develop experimentally testable hypotheses and so forth that otherwise would be difficult to imagine. And so we've been working in my group for some years in various diseases like influenza, cancer, chlamydia, and trypanosomiasis, and have had a sort of a growing body of experience in applying these methods to actual problems in health and disease. And so just a very high level background slide. So presumably, I would guess most of you know what all atom molecular dynamics simulations are. But basically what we're doing is we're taking these biological systems and where we're getting data typically from X-ray crystallography and cryo-electron microscopy. And I'll tell you a little bit more about those techniques and the data we can get in a little bit. But we take this experimental data that gives us the relative position of all the atoms in our system. We then approximate the atoms in our biological system like hard spheres, and then they are assigned a particular charge and other sort of features that come from the so-called force field. And then we have this potential function here, which is relatively straightforward. This potential function describes the different interactions of our system. So every 2 atoms that are connected form a bond. You can see the term there. Every 3 atoms that are connected form an angle. We have a dihedral term. This is 4 connected atoms. And then we have our so-called non-bonded forces where we have van der Waals and electrostatics. And then all we're basically doing is integrating Newton's equation of motion over time. So we start with one particular configuration that we get typically from experimental data or experimental data sources, multiple, as I'll show you for these sort of larger systems. We start with some particular configuration. We integrate one time step. We get another configuration. We integrate another step. And so forth. And we do this integration millions and billions and trillions of times. And that's how we basically generate sort of a propagation of the atoms over time. And we have these so-called ensembles of structures and sort of can start to explore the dynamics of these systems. And this movie is not going to find, that's so terrible. This is supposed to show you sort of what we see, the so-called the wiggling and jiggling of the atoms that we can see. I'm sorry that the media is not found. That's because I threw this talk together at such last minute. But basically, so why do we care about doing this? So, the reason why one of the reasons why we do these computational approaches is because the experiments, whether it's crystallography or cryo-electron microscopy or other sort of imaging modalities, for structural biology, typically they're able to get us a nice high resolution structure. But in chasing this sort of high resolution structure, oftentimes they have to sort of perturb the system in order to dampen the dynamics so that they can actually resolve the positions of the atoms successfully. And so this could be introducing particular stabilizing mutations. It can also take the form sometimes of truncating loops that are particularly mobile or truncating completely off particular domains of a protein. And then, as these they're done at very low temperatures, or so it could be also cryogenically frozen. So what we get from the experiments is just a static structure, and that's very informative, of course, because just even knowing the relative position of the atoms, you can already start to tell a lot about the function and the properties of these different molecular machines. But the reality is that these are highly dynamic, mobile, moving machines. And so what we can do with these computational methods is to predict using the laws of statistical mechanics. So in a theoretically rigorous manner, we can begin to predict the motions and the thermodynamic and kinetic properties of these systems. And the advantage here is that, of course, it looks in some ways just like a video game or like computer graphics. But of course, because we're doing this in alignment with the laws of statistical mechanics, we can actually extrapolate out experimentally testable macroscopic observables, right? So we can refer back to experiments and in a way that is rigorous. And so so what I'm sorry, this movie doesn't show you, it's because it should be wiggling and jiggling, but basically what we see here is sort of the fluctuations of the motions of the atoms over time. And what we often see is actually that if we just looked at, for example, the crystal structures, and this is a protein that I'm showing, it's called P53. It's a cancer-related protein. If we looked at all of the crystal structures that are available experimentally in this one pocket here, which is of interest for drug discovery, what you see is that this pocket is relatively small and one of the residues is sort of tucked back and occluded from solvent. And what we see happening over just a few tens of nanoseconds, we actually see this pocket relax into a more open conformation. We see solvent or water molecules going in to this site and sort of helping to open it up. And so this is a so-called open structure that we're able to sample from the dynamics. And why this is important is because these sites in these computational simulations, they can open and then they can be druggable. And so with knowledge of these sites, as predicted from computing, these sites which have never before been seen in crystallography or other experimental techniques, generally because they've frozen out the dynamics, now we have an additional edge on the design and discovery of small molecules. So this could be important for, the treatment of disease, for example, or understanding cryptic epitopes. These pockets that aren't seen, these new sites, they sometimes refer to them as cryptic because they're otherwise hidden. So that's a term that you might also hear being thrown around, particularly with SARS-CoV-2. They and many infectious diseases, they're also concerned not just with trying to find these novel druggable pockets that can open up, but also trying to figure out how mobility in regions that are recognized by antibodies, how mobility can change the structure and ultimately the recognition of like vaccines and so forth. So there's a lot of interest in trying to develop these models and methods for these systems. Okay, and so what we would like to do, in general, so what you're looking at there, this little blob that looks sort of, I don't even know, like a liver or something, that's actually a ribosome drawn it by David Goodsell. And, typically computational folks and also experimentalists, they tend to, you isolate your system. We take this sort of reductionist approach, right, to biology, where you express your protein, and then you concentrate it, or you put it in sort of these sort of fictitious solutions so that you can do all sorts of manipulations and get the structure and so on and so forth. And this is, again, really, really informative. But what we want to do, and we do the same computationally, where we take the protein and we basically just put it in like a drop of water with ion buffer around it. But what we really want to do is understand how these machines actually work in their in vivo environment, right? Because at the end of the day, for example, if you do want to develop a drug, you're putting it in a human body. Vaccines, same. There's so many elements of complexity that you can't necessarily access in particular sort of experimental modalities. So we have been using, we and others, take the approach of trying to use computational biophysics, essentially, or different types of biomedical research computing to help sort of understand the phenomena going from the molecular all the way through to the subcellular, cellular, tissue, and even whole organ levels. And the only way to really do this is generally by using multi-scale approaches, which I won't go into too much detail about, but it's just to say that we are in general really good as scientists at sort of like working within one particular domain, right? So we have many tools that inform on the molecular and macromolecular structure. This allows us to access particular spatial and temporal scales. We have other techniques at the subcellular range and other techniques for cells and so on. But there exist these so-called technology or capability gaps that are sort of dark, in the sense that it is very hard to understand the molecular machinery with all of the details that one needs to design chemical matter, which at the end of the day are very small atomic interactions. Very difficult to do that when the molecular machine is inside its fully elaborated subcellular compartment or its cellular compartment. So we've been working to try to develop new tools and techniques that allow us to go into and across these key capability gaps so that computing can help sort of see the unseen, right? Or the inner workings of cells at the molecular level. And so this talk is going to be very high level, and I'm not going to get into too many details, but I just wanted to, if people are interested in learning more about particular methods for multi-scale, I just wanted to point out to this review that I wrote with a colleague, Adrian Mulholland, now a couple of years ago, where we talked about the development and application of multi-scale methods, particularly with the focus on how they're useful in bridging chemical and biological complexity with an eye for drug discovery, if you want more information. But so we've been working to try to move beyond sort of a single. Oh yeah, ask questions.
Can we chime in and just, so how much do you
do it? Yeah.
How do you like to do it? I don't want to be rude, even though I just want.
No, that's okay. Just go on. Let's talk. Yeah, ask.
So how much do you interact with other fields on this? So you're simulating physics and chemistry at different scales. And I'm just wondering if there are certain techniques you use that, like, are there standard techniques you use? Do you discuss these with other fields? The reason why I ask this is because when I first saw that, the first thing I thought of was exoplanet formations. This is how planets form in solar systems, because at that point you have like tiny bits of dust, and then you have to start scaling up. You start out with just, you start out with atoms, and then you end up with like complicated materials. So you have these, the same type of transitions. And then all of it, of course, matters for their simulations, which is its own sort of nightmare, which sounds sort of similar to this. And I'm just wondering if, I don't know, if this is like its own field of study on how do you do this multi-scale.
Yeah, that's a great question. That's a great question. I love multi-scale. So I would say, yes, the sort of the tyranny of scale is something that, we see everywhere in science, right? For planet formation, for understanding ecosystems, for this type of stuff. I think there are parallels. Certainly there are parallels that can be drawn. Methodologically, one issue is that when you cross scales, you do need to change the interactions and the physics underneath each of those scales, and that the challenge then comes really at how you sort of do that handshake across. There are times when we do, like, for example, when we're for the biological multi-scale stuff, we have at times it is helpful to to talk with other people who are doing sort of modeling of totally different types of systems, and I think that there are typically, so I would say, to answer your question just without talking super long about it, yes, we do interact with other fields, and we can take inspiration from them, but at the end of the day, exactly how much can be sort of brought in directly from a method of a different field, sometimes things have to change because the properties of the systems are somewhat different, and so on and so forth. So I think, yes, it's good for inspiration, and because the challenges sometimes are similar, but it's not always a direct correspondence, obviously.
Okay, thanks.
Yeah. Okay, and so we've been trying to, we've been very interested to try to build essentially 3D, to try to take advantage of the sort of enormous data growth that we've seen in structural biology because of advances, various technological advances that that have come into that field. We've wanted to take that data to basically try to build what we call 3D visible virtual cells, essentially. And so, the idea is, again, in trying to go across scale, is that we have these different structural techniques that allow us to gather data essentially at different resolution with some different constraints. So with electron crystallography, we can get very detailed structural information about like one particular molecule. With approaches like single particle analysis or cryo-electron microscopy, we can then begin to understand, okay, how these singular components, how they actually interact within their macromolecular complexes. And then going to the next stage with using electron tomography with focused ion beam milling, they can basically freeze these cells, take a very thin slice, and use tomography to basically understand how these macromolecular complexes are situated inside of their subcellular compartments. And then we can switch imaging modalities to what they call resin embedded samples and get, again, to sort of higher scales of organization, where, you know, out here with serial block EM, basically they can put entire tissue samples inside of the microscope, and then basically with a diamond knife they can they image one layer of the sample, they shave off a very sort of thin slice, they image again, they shave off another. They do this, these microscopes run for days and weeks at a time. They generate huge data sets, you know, petabyte scale. And they have, at the end of the day, hundreds of thousands of these tiny molecular structures all inside this sort of new mapping, you know. So there's a lot of work at each scale, but also what we're trying to do is develop tools. What we've been doing is to develop tools and techniques that allow us to sort of integrate this data across scale so that we can do things like, I'll show you what we did for influenza, where we've moved from studying a single protein, you know, in a sort of dilute solution to actually trying to construct, there we go, to actually constructing, like, for example, the entire influenza viral envelope. And so one of the things that, so we've been working on for years is developing tools that allow us to make this new mapping from the single particle to like the whole virus level. And so this is not SARS-CoV-2. So until, geez, about 2 months ago, we've been working for years on modeling the influenza virus, which has some similarities to SARS-CoV-2, but it's a different type of virus. This is what we built. So this is using these integrative models, we combine data from high resolution X-ray crystallography with lower resolution electron tomography, together with different sort of molecular modeling tools to build this this viral particle. This had about 160 million atoms. It's fully solvated, but I'm hiding all the waters because otherwise you don't see anything. Influenza has a host-derived lipid envelope, which is very similar to what we see with SARS-CoV-2. And so we were able to simulate this virus on the petascale Blue Waters machine, which probably many of you are aware of. And possibly have used. So, you know, you can only run these systems on, because of the size of these systems, and this is among the biggest biophysical systems that has been run yet on these machines. You have to use these very, very big architectures because the code that we're using, in this case for the big systems, we typically use NAMD2. It basically will run on hundreds or thousands, in this case it's typically thousands of nodes on these petascale machines. And so you can see that I'm sort of translating and rotating this viral particle to sort of give you a little tour, but you can also see the really sort of small, detailed, and here we're inside the membrane, but we're gonna back out. You see the little motion of the atoms. That's the wiggling and jiggling of the atoms as predicted by the molecular dynamics. That's where we can derive the time-dependent properties from. And so I'll just say too, it's not just that we care about necessarily the movement of every single atom. Sometimes we want to go out, as I was saying, to like longer time and length scales. And so we can do that by using the same model, but switching the physics. So in this case, you know, once we've built these systems, or have a method to build these systems, we then can, instead of running necessarily all atom MD, we can also use things like Brownian dynamics, where in Brownian dynamics, we're freezing all of the internal degrees of freedom. So the objects basically move like rigid bodies. And then, you know, they're sampling Brownian motion, moving through a solution with a particular viscosity and subject to like electrostatic properties of the field, essentially. And so that allows us to go out to longer time and length scales and to start to ask different questions. It's also computationally much cheaper. So in the case of influenza, what we did was we were able to construct multiple variants at the whole viral level. We could actually construct multiple variants of different strains where we're changing out the different pieces, which viruses do in real life. You know, they change out different segments as they mutate, or they can adopt particular mutations, and then we can interrogate them in different ways. And so, like for one example here, you can see in the influenza virus on the left, you see the blue protein and the white protein. These are the two glycoproteins on the surface. We can, on the right here, you see this, the lighter green protein is actually significantly shorter than the white one here. So that's we can basically introduce these types of mutations as seen in nature. So this stalk deletion is something that comes into play for the bird flu, which is highly pathogenic. And then we can interrogate these systems and try to, again, sort of drive experimental design. So, because we had been working for so many years with influenza, I think it has allowed us to sort of pivot very quickly into SARS-CoV-2. And so our publication with the simulation of the flu virus actually just got published on February 19th, which was really like about the time that it was just a few days after, in fact, that we started to pivot our efforts very seriously, as I'll tell you about, towards SARS-CoV-2. So, this paper came out, we were very happy, we got one of the covers. And then, in the background, all of us, I'm sure, including you all, we sort of had this more and more thoughts about the SARS-CoV-2 virus, about COVID-19. And so I'm just showing here the date and the cases in logarithmic scale, that people saw in different countries. And so it was right around this time when we started working in SARS-CoV-2. So as I was mentioning, this is all stuff that we weren't necessarily working on before, even though we had worked on sort of similar challenges, but not this particular system. And actually, the day that we really pivoted was the day that the cryo-electron microscopy structure of the spike protein in the prefusing conformation was posted into the bioarchive by the McClellan lab at the University of Texas, Austin. So this happened on February 15th. This was really when we saw this drop and we said, okay, maybe we really need to take the SARS-CoV-2 a bit more seriously. And so we started exploring the system, because for what we do, as I mentioned, it's very dependent on having a good initial starting condition, or initial boundary condition, if you will, for your system. And so ideally you want to take that from experiment. So until this paper came out, everything was just sort of homology modeling, which is like, we know something kind of similar about a related protein, but we don't actually have the data for the particular one. So this was a phase change in terms of, for our field, how we could begin to explore and interact with these systems. So just sort of a brief overview. Apologize for the graininess of this particular image. This was done by a medical illustrator named Veronica Falconieri. Her credited up there if you want to get in touch with her. But so the SARS-CoV-2 variant is shown here on the left. You can see it looks a bit similar to influenza. It has a host-derived lipid envelope. It only has one type of glycoprotein on its surface, and it's a so-called spike. They call it the spike protein. It's significantly larger than the proteins that are on the influenza virus, and it has significantly more complications, I'll say. In particular in the form of glycosylations. So these viruses have these spikes sort of sticking out of them, and these spikes are really the infection machinery, if you will, of the virus. They contain, they're trimeric, so there's 3 different homodimers that come together to form this complex, but it's even a little bit more complicated than that, because each one of these monomers has different segments to it. And so what they've been able to resolve in the cryo-EM is this top bit here that you can see with it actually has some sort of structure to it. They, as I was saying, in order to resolve this, they chopped out, they had to chop out particular bits. They chop out this whole, basically, stalk. There's a stalk that tethers these infection machinery spike protein trimers basically to the viral membrane. And so that's why this is depicted here as really a cartoon, because they don't actually have resolved structures for that. And so these, these spike proteins, basically they sort of, they hang off of this, the viral particle. One of the interesting things about SARS-CoV-2 is that it has picked up features that people haven't seen before in SARS. So one feature is this, there's a site between the 2 different purple domains on each monomer. It's a so-called furin cleavage site. And it's believed that this was some sort of new evolutionary feature that it's cleaved by a protease in the host cell. So protease are basically like these scissor proteins that like go all around your body essentially, and they're cutting up protein bonds within like a protein segment, and it sort of cleaves this site and actually maybe allows for more mobility, and that could potentially be something that has led to like the increased infection in humans. But this is something that, you know, people are really just starting to experimentally and computationally explore. There's a secondary cleavage site here. This is the S2 prime site that also has something that also must be cleaved before like the infection, before it can be it can sort of go into the host cell. Okay, so the paper came out with the cryo-EM structure. We started to take a look at it seriously. We said, you know, I bet we can probably do something really quickly, or we can try to do something quickly, but also of course we have to do this carefully. But we're going to need compute time. So actually, I called up Dan Stanzione at the University of Texas. He's director of TAC, or the Texas Advanced Computing Center. They had recently been awarded and built Frontera, which probably many of you know of. This is the 5th fastest machine on the top 500 down here. And I said, hey, Dan, you know, this coronavirus isn't looking too good. You know, I think we would like to explore it. You know, I know that because they had an open call for proposals and it was basically due like in 2 days. I said, look, we want to do something, but I might need it. Is it possible to get a little extension so that we can start to explore it? And he said, he basically said, yeah, Rommie, we know what you do, like, you know, go for it. So I have to say they've been tremendously supportive. There's actually a big consortium of HPC, you know, computing now that is responding to calls for compute time for various groups all around the world to look at different aspects, including for epidemiological modeling, which of course takes a lot of their time. But I would just say the kind of work that we do is really enabled by these machines. And I think the support of Frontera in particular, I just wanted to give a shout out to, because we wouldn't be able to do this work without them. Okay, and so our approach, again, sort of taken off of what we had done with influenza, you know, we started looking at these, at the data that was out there for coronaviruses and for SARS-like viruses in particular. So, you know, they're called coronaviruses because of this particular arrangement that when you look at them, the spike proteins, so this is the virus, the spike proteins basically sort of stick out on the surface and it looks like a crown, you know, the top view of a crown. So that's where the name comes from. And then so our approach is to, again, sort of combine data across scales. So what we're aiming to build is a particle that has a membrane diameter of about 85 nanometers. And we know this from the tomography experiments. And then we know also the stoichiometry of the spike proteins, again, based on the tomography and various experiments. So in this case, the diameter of the membrane itself is about 85 nanometers, and then the spikes each stick out about 25, you know, for an additional sort of 50 on either edge. So all told, this virus will be a little bit larger actually than the influenza virus we've already simulated. And then we start to so what we need to do though is, before we can sort of build into the system, our approach is to build really good models of each of the components. And so the first component we really have started to address, which is the one, pretty much the only one I really talk about today, is the so-called spike protein. And that's because they have a number of really beautiful structures already resolved, both with cryo-electron microscopy, as well as with smaller pieces in really high resolution X-ray crystallography. Sometimes actually these pieces come actually together with the human, the part of the human receptor that the viruses bind. So these two proteins here that I'm showing in D and E, this is just the virus protein, or the viral protein, the spike protein. And then this last panel F, this yellow blob here, this is the ACE2 receptor. So how it works is that this virus is basically, it comes into, it sort of begins to approach a host cell, and it's interacting with the so-called ACE2 receptors on the host cell. So your host cell has all sorts of stuff sticking off of it. And it's sort of a recognition or a handshake that happens between the spike protein's receptor binding domain, which, by the way, in order to make that handshake, it has to sort of stick out its hand. And that's what it's doing in this so-called up conformation that I'm highlighting here. They also have resolved the structure in the down conformation, where the hand is sort of inside, but it opens up and then can actually make the interaction that actually infects. And so we're interested to understand. So people like me and like many in my field, we're interested to understand what this handshake looks like, what this interaction looks like, because if we do, we can potentially develop new therapies that block that interaction from happening, for example. But there's many different types of modalities that one can think of trying to design for therapeutics. That's just one, but there's many insights that computational methods in fields like this can give to the design of new drugs, or also the understanding of antibodies and so forth. Okay, so what we need to do is we have some of the information, as I showed you, from experiment, but we don't have all of it. And so we need to fill in the gaps. And so to do that, we do that very carefully, because this is where we have to be more, we end up being more speculative, because we don't have direct experimental data to build off of. But in many cases, we can create sort of reasonable models, and we think we've done that with the spike. So the spike protein has, this is the whole sort of, this is one chain sequence, right? So these proteins have sequences. So I'm showing sort of the end-to-end sequence. It has different parts. So it has first this N-terminal signal peptide that's typically cleaved off. We don't include that. Then you have the S1 domain, which I showed you before in purple. Highlighted, overlapped in pink is the so-called receptor binding domain. That's the bit that basically sticks up and makes that handshake with or recognizes the receptors on the host cell. At the intersection of the S1 and which I'm showing in yellow, and the S2 domain, which I'm showing here in green, I mentioned there was a cleavage site, a furin cleavage site. That cleavage happens right here, right after position 685. In white here is a stalk that basically, these glycoproteins on viruses often have this sort of like, they're connected in some way to the viral membrane. And so in this case with the spike protein, it's just sort of like a long helical bundle. And we had to model that. And then there's a bit that also goes into the viral membrane. That's the transmembrane part, which is also basically a helix. And then this last bit here is an intravirion tail. And it's got some cysteines that actually are palmitoylated, which are these sort of like anchoring groups that kind of keep it like a foot into the membrane. So we're trying to model all of that. The other thing that we have to worry about that you might hear people talk about on these spike proteins is something called glycosylation. So glycosylation, they're basically like sugar molecules or sugar-like entities that are used for a number of different things inside cells, very often with recognition and they can take basically different forms and have different sort of compositions. So yeah, without getting into too much detail, the SARS-CoV-2 spike protein. So what I'm showing here is an alignment of SARS-CoV and SARS-CoV-2 and looking at the different glycan sites. So on the spike protein, actually, which I showed you here, but I didn't talk about it. So if we go back to the structure of the cryo-EM, these dark purple bits here that you can see, those are actually the locations where these glycans are, or these sugars are. So these spike proteins actually have essentially like a coating, and here you can see them too. These are all like the darker, little dark stubs. They have like essentially like a coating of sugar, and so these they have different purposes, but you know, the main thing is that it helps sort of shield these viral particles from detection by the host system, because they basically can adopt sort of the same sort of camouflage shell as the host cells themselves, right? And so that's one of their features. So, but the thing is that experimentally, they know where these sugars should be. And in the cryo-EM, they can actually see a little bit of the sugar. They can just see like the first, usually 1 or 2 or sometimes 3 residues, but it might actually be much more complicated than that. And that's just because there's too much like conformational heterogeneity. They're too flexible for them to resolve in these sort of static structures. So we're trying to build them back into our models, right? And so we build them in at these different positions. And so one of the interesting things about the SARS-CoV-2 is that it has 22 glycan sites on every monomer, which is a lot, actually. It's a lot more than the number of glycans that we see on other viral glycoproteins, for example, like what we saw with influenza. So these are really complicated, heavily glycosylated systems. And so what we've been doing is basically trying to build that back in. There's a number of groups who have, there's this paper, there's 2 other really good papers that actually did a site-specific analysis of at each of these sites, like what is the composition of the glycans. And so this is what this mapping is showing. And so we actually build that back in. So what I'm showing here is sort of this is like a legend or like our mapping when we're building the structure. These are basically these additional sort of chains here that I'm showing. These are the additional chains that we built in at each one of these sites for each of the monomers in the spike protein. And these basically are essentially experimentally unresolved. They're unseeable, essentially. But we know they're important, and that's why we want to include them. And I'll show you how striking it is when you include these, how different it is, and if you look at just sort of a naked unglycosylated protein. Okay, so in addition to the glycans, we also need to fill in the other bits that are missing. We're missing some loops. So as I mentioned, sometimes the loops in these sort of static experimental structure resolution methods, they can't, if the loops are moving too much, they don't get good enough density. And so they don't see them. So what that means is that when you look at the data they have, it's just like there'd be a vacant spot where there should be some kind of connection. There's nothing there. So we have to model those back in. And then the other thing that we wanted to do is actually, we want to build, essentially, we want to get to the point where we can have the whole viral particle. And so not only do we need the stalk domain or the trimer head that they resolved with cryo-EM, but we also need to develop the tether that connects it to the membrane and the viral membrane itself, et cetera. And so we do that by homology modeling. And so that tether bit that sort of connects the stalk, it basically looks like what we call a helical bundle, but there's some insertions of loops there that sort of give it a little bit more flexibility. Okay, so this is what the whole structure looks like when it's built. This is what our model looks like. And so on the left, I'm showing you the closed spike structure. So this top bit, so if I drew a line at about like the median here, this sort of like at like about halfway and up, this big bulbous spot part, that's what they can resolve experimentally. And everything south of that, we've basically modeled using our methods. So this is the long stalk, and you can see the membrane. Okay, and then also we have it in the closed conformation and the open conformation. This is what they see. The other thing that we are doing is that the membrane, we've sort of, the membrane is important. It's host derived. So, and these guys actually, the viruses actually bud off of something called the endoplasmic reticulum and the Golgi apparatus, which basically is sort of some compartment there. And so it has, the membrane that we actually have built is sort of ER and Golgi-like. And then this is the transmembrane bit that I was saying. We also had to model that. And then there's these sort of intravirion tails that have cysteines with that have been palmitoylated. And so what basically that does is like, it's like little feet that just kind of like go, they snorkel up into the membrane because they're pretty hydrophobic, and they sort of anchor that guy into the membrane. And so what you're seeing here, so the different pink colored bits, or the stripe across the screen, that's what the membrane, kind of like a depiction of all of the different lipids and lipid-like molecules, including cholesterol, that are in the membrane. You have the transmembrane bit, and then this is the bottom of the stalk. And these colorful balls here, this like yellow and blue and green and purple and red, those are glycans. So these glycan groups that we've built off. Okay, so on the left is basically what the fully glycosylated spike looks like in one atomic representation, right? So all of this, the gray bit underneath that you can see, that's what they were able to resolve experimentally. And pretty much, and then they got stubs for each of these little sites where we've sort of built off these sugars. And what you see just by looking at it is, I mean, at least for us, it was just like, wow, it's really covered. It's really covered top to bottom in with these glycans. And so here, finally, I have a working movie. I'm sorry that my P53 movie earlier didn't work. But this sort of wiggling and jiggling of the atoms, that's the data that we get out of our molecular dynamics simulations, okay? And so we're looking at sort of the femto, in this case, it's nanosecond type time scale dynamics of how this machine is moving, and you can see the lipids moving. So this was originally, all of these lipids here, this was our original model, or one of the early models. So you sort of like kind of build up you sort of continue to sort of build up and sort of like refine your model as you go. Initially, we just had what we call POPC molecules in the membrane, but we've since made that more realistic. And then I want to show you this picture too, because this to me is just so fantastic. So what we're showing here in blue, the dark blue sort of like mossy lines, this is if you look at all the positions that each glycan, so if we go back here and you saw them moving around, if you represented these like lines and you sort of trace their movement over time, you end up with these little, each of these glycan sites basically makes like a little, it looks like a puffball or something, but it's because it's moving around the structure. So this is actually what they refer to as the so-called glycan shield. All right, and you know, they call it a glycan shield, as I mentioned before, because often when we think about the glycosylation of biological systems, it is sort of like a protective layer of sugars, and especially for viruses, by having this coating that's very similar to the coating that's on the host cells, then our immune system doesn't detect it necessarily as being anything bad, and so it can like go around kind of in camouflage and you know, and get to where it wants to go. And so but I think what's so striking to me about this is how different this glycosylated protein looks like compared to just the naked protein. And I say that because a lot of times people will develop models completely in the absence of glycans, because they add a significant additional layer of complexity and pain in terms of the modeling, and also with force fields and so forth. Although the force fields have been worked out, I think, pretty well for the glycans. But you can see that if you have a model that doesn't have all the blue furry bits, you're going to have something that looks completely different, and something probably that is not going to be as biologically as predictive as if you include it. And so this is looking again at the full spike protein with the glycans. This is looking from the top down. Okay, so this is in the closed form, what the host, if you were, you know, what the host cell sees. So you mostly see sugars, you mostly see the sugar bits. In the open conformation, something that's also quite striking is you see there in the open conformation how the receptor binding domain is up. And by being up, now it's lifted out of the glycan shield, which is just, again, sort of, for the kind of stuff we do, I think this is super cool, because it basically tells us, you know, essentially like why this thing needs to move up into the open conformation. You know, it's partially because to make that handshake, to make the recognition of the infection happen with the host cell, it's got to expose that hand or that piece of the infection machinery. And this is looking, again, top down. You can see that in the open conformation, now this is a much different picture that the host cell sees. And this is again just from the side. So this is in the down conformation. You can see that everything's sort of hidden from the host cell. And in the open conformation, that infection tip is sort of out and it's revealed and it's ready to do its business, so to speak. So that's sort of where we are. We're doing a lot of analysis. This is the full system. It has all of the glycosylation. It has a few cosylations. Some of the bits they have also sialation sites, the full membrane, etc. And as I mentioned, we're basically working on not just trying to explore the time-dependent dynamics and different bits that are exposed and unexposed and make, you know, insights into just the spike, but what we really want to do is sort of go to higher levels and try to model, for example, what the whole virus looks like. And so, you know, we're working towards that. You know, and eventually, you know, what us and others would like to do, and this is another sort of rendition from a medical illustrator, this one from the Cognition Studio. You know, what we would like to do is actually have in silico models of very, you know, multiple in silico models of the virus here with multiple spike proteins, and then actually have, you know, models of a host cell where you know, you can have the so-called ACE2 receptors actually also fully glycosylated and other things on the host cell, so that we can begin to understand molecular recognition and so forth. And there's many people working on, you know, various aspects of this problem. But these are the kinds of things that we can look at and we can do and explore with these methods. Okay, and it's 10:57. Oh my gosh, I hear like a little note, so maybe somebody's like trying to tell me, Rommie, stop talking. But I just wanted to also say our field, and maybe I should look because I hear more like little, is somebody trying to say something?
No, I don't think so. Maybe there's some background.
Oh, okay.
Please continue.
Okay. I'm almost done. I just wanted to say, and as I mentioned in the beginning to Peter, you know, one of the things I've always admired about particle physics, and sort of I think where many of you work, is how, you know, you have really driven the development of these large computing architectures and you know, and these amazing data sharing networks and, you know, equipment where, you know, you have sort of one data set and you have the whole community, you know, working on this same data set in a really sort of beautiful, cohesive way. I have to say, I think, you know, that's something where in the biosciences we are very far behind in terms of, I think, how we work. You know, it's much more these little fiefdoms, is sort of very rogue. So one of the things that we did with SARS-CoV-2 is, you know, we have developed this worldwide sort of worldwide collaboration and sharing agreement. There's over 200 different groups that have signed on saying, okay, we're going to try to do this openly. We're going to try to share. So our models are going to be available and so on and so forth. And I think it'll get us a little bit closer to where you guys are, but obviously we have many generations to go before we have anything close to how you've done it. But I'm very excited about that. And also the NSF MOLSI, Molecular Sciences Software Institute, together with BioExcel and CCP Biosim in the UK and sort of other consortia all over the world, we're trying to develop a sort of what they're calling the COVID-19 Molecular Structure and Therapeutics Hub, which will aggregate all the critical information needed to accelerate drug discovery and molecular simulation and modeling, including our trajectories and our simulation data, but also information about small molecule drugs and antibodies and so on and so forth, organized by targets for SARS, all within SARS-CoV-2. So that's also another sort of exciting aspect of working on this problem, you know, in real time, that I think I've been very excited and sort of heartened at people's willingness to, you know, collaborate and to share. This is something that, you know, people in general can be good about collaborating and sharing, but this is like taking it to a new level for us. So I think that's of course very good. All right, and so all in my talk there, I'm happy to take questions. You know, I'm sorry, it's already at 11 for me. It's very late for some of you in Amsterdam. I just wanted to acknowledge in particular, so folks in my group, of course, Lorenzo Castellino and Ziad Gayab literally probably have not slept very much at all since February 15th. They have been working around the clock, and a whole team of folks, including also other theoretical and a number of experimental groups. Michael Feig has been helping us build different parts of the system. Ben Newman is an M protein expert. Again, I wanted to thank Dan Stanzione and the Tech Frontiera team. They've been fantastic. I said thanks to Michael Feig at MSU. Simon Khalid from Southampton is also helping us, and Elisa Fada from Dublin, along with others who are also collaborating with now. So I'm happy to take questions. I hope that was somewhat understandable.
That was excellent. Thank you very much. So I will open it up to questions from others. You don't need to do the formal hand raisings. People should come forward.
Yeah, I'm
Oops,
trying to
I was
see.
muted. Thanks for the great talk. It was very interesting. I was curious, and we could kill the recording, but like, what are the big challenges you face? Because one of the things that surprised, we spoke with someone from Folding at Home, I don't know, a few weeks back, and it sounded like a lot of the challenges with this very fast corona response wasn't always like methods, because it was just about throwing some amount of computation at the problem, but that the cracks were more on the organizational side. I was wondering, I don't know, like when it actually comes to you guys doing your work with all of this extra pressure on you, like where do you see the weaknesses, I guess, if that's a fair question?
Yeah, that's a fair question. You know, I think for us, I mean, you mean like as a field or for my group?
I more meant as a field, but I'm also curious. Honestly, I'm interested in both. So
kind of both. You know, from a field perspective, I think the sharing is something that is sort of like, we know we should do it, but it's, you know, it's challenging. Also, the reward system that we have, the incentive system, is not necessarily conducive at all to sharing. And, you know, it sort of incentivizes the wrong thing, particularly in this case, you know, where we really kind of want like real time, and we can't necessarily like do this whole song and dance with, you know, making a pretty publication and going through review. It's like, well, gosh, if that's going to take 8 weeks, I just need to get this information, you know, out there. I think one needs to be careful, one needs to be very careful to not just throw a bunch of computing time because you have it. And we do have enormous amounts of computing time, but one has to be very careful in developing a model that is accurate and that is good. And that A, takes a lot of skill, B, it does take time, C, at the end of the day, you don't really know how good your model is until it's put to test experimentally, right? So that's probably the most challenging bit is, I would say, probably connecting, for many people in the field, it will be connecting to experimental groups in a meaningful way, and in a time scale that will be amenable to, you know, making a discovery that actually will have impact. You know, I would say you know, my approach on it is get in touch with the experimentalists early and often. And so, you know, we're talking with Jason McClellan, and we're talking with experimentalists all over the place, because, you know, I think that's going to be where the ultimate test of, you know, how good any of these things are is, you know, at the end of the day, how the models have been used to actually have real impact, which is not a paper, even in Nature, you know, it's like, what are we going to learn that's going to change the way that people think about the disease, or that they treat the disease, or, you know, can it improve the cocktail of antibodies, or, you know, et cetera. Because of course, if you look at like
experimentalists, are they other academics, or are they people in pharma?
They're both. They're both. They're everywhere.
And how do you communicate your results? How do you publish your data, as it were?
I mean, ordinarily we would, you know, keep everything in and do this analysis sort of very slowly. And then when we get to a paper, you know, then you kind of, you know, you drop it in the archive, and maybe that's when you tweet about it or whatever, or when it's published, you know. But in this case, it's
I guess my question was not about the sociology of it, but just mechanically, what sort of data do you share? Are there well-defined data structures and data formats that you use in your field and that everyone agrees on, so you can simply upload it somewhere?
Yeah, I know.
How is the data exchanged?
Okay, so data exchange inside the field, you know, I'll say, unfortunately, is not something, I mean, it's something that we're still working on. So there's sort of like maybe 4 or 5 different camps of field types. or file types, I mean, where people can like easily exchange within each camp, but then like cross camp exchange like becomes a little bit harder. So we're working on that. I mean, things are mostly agreed on, but there are these just like little differences, and sometimes the devil is in the details, with really making like a fully correspondent, sort of exchange. Then with experimentalists, it's different. They don't care about our technical input files. They want to know at the end of the day, what residue do you want me to mutate, or what drug do you want me to make, or how should I change my antibody? And that's a whole different set of skills in terms of communicating to these groups, which is also a problem, I would say, or a challenge for people in my field. We tend to be sort of narrow. And it's like, usually it's a different set of folks who can speak and work really well with experimentalists, because it's just like a different way of, communicating and like acting on the data. So there's multiple aspects of it, but there's not one like agreed standard for everything in the field of even just like molecular dynamics. No, it's been a problem. I would say that's been a challenge for us. And places like MOLSI, this Molecular Sciences Software Institute, are trying to get everybody to a place where we can sort of have interoperable, workflows and units and this kind of thing, but it's still a little bit tricky. Did that answer your question?
It addresses the question for sure. Are there specific technical computing bottlenecks that are distinct from the domain knowledge? Because are there places where if you had outside help, you think someone might be able to speed up your code by a factor of 2, or that if you had people available who are not domain experts, they could help you with finding ways to change your data format into someone else's data format?
Yeah, I think there is possibly room for help there. That is some, again, that's sort of like activities that we need to do for sure in the field. People are sort of like reluctant to do it because they just don't want to necessarily, I think, put in the, the effort, but maybe now would be a time to potentially get help from other people in the field. As far as like speeding things up, I think that's sort of a longer conversation. That part is less clear to me exactly if there's, particular bottlenecks. I think, maybe on the multi-scale question, yes, not necessarily like within one domain. I think those problems have been pretty well studied and sort of worked over, but possibly with multi-scale. But the answer to your second question, if there are people who are technically competent who want to help, I would say, I think probably getting in touch with the NSF MOLSI as we sort of try to, as they're working on trying to make things more interoperable, where it's not that you just need to make like parsers, but it's almost kind of like that, like a lot of sort of interconversion things. That could be an area. The other thing that we're trying to do, but it's less technical. We're also trying to get like domain experts in on each of sort of the targets for gathering information and so forth, but that's not what you're asking me. But it's like another aspect of the challenge for COVID.
Well, that's actually a really interesting question about how do you build a team of people who maybe are used to competing with each other?
Well, this is the thing, right? I mean, ordinarily we kind of are competing with each other in this sense, but this is a different beast, right? So I think that most people, like 99% of people at least, are saying, yes, let's work together. They sign their names to this thing saying, we're going to share, we're going to be open, we're going to collaborate, we're going to work together on the same data. And I'll say, even for myself, so the Spike model that I showed you, we've now shared that model pre-publication, which kind of freaks me out, but we're going to do it. We've done it, we've shared it with like 6-8 different groups, and they're taking it and they're working on it and they're converting it to their own sort of sets of requirements for their methods, and so we're going to see. This is a real life experiment also for the field about, you know, what happens when we sort of turn things on its head, on everything on the head, and try to work together. I'm hoping that actually, because I've long felt that, like I was saying, I think we need to be more sort of community oriented, like you know, what you have in physics or what we see in physics and in other fields. And I'm hoping that this will sort of help that process, but I guess we're going to find out.
So questions from others? So I have a question. So you're using Frontera, which is got a particular choice. As you may know, there's on the DOE side, there's the leadership class machines like Summit that are very GPU heavy, but that's not Frontera. So, you know, why that choice, and why, you know, are the codes not amenable to running on GPUs, or?
No. So, okay. So, actually we have, so it kind of depends on the system. It depends on the system size. So for most of what people do typically in my field, which is like study a single molecule, actually you can, there are codes like Amber, GPU, and GROMACS, and there's a number of different codes that just run pretty much like on a GPU alone, and they're super fast. Like they're really great. But the challenge for us is that these systems are getting to be pretty big. So even just the Spike, because we have like the whole tether and the membrane, it's about 1.7 million atoms, which if you just try to put that on a single GPU, it's not very good. So like our systems don't map well to the folding at home type of like many distributed computing bits, because actually we need to, so one of the reasons why we choose Frontera is because for the larger system sizes, so the Spike being like, you know, 1.7 million atoms, when we go to the whole SARS-CoV-2 viral envelope, it's going to be like 200 million atoms. You have to be able to like run it across like many hundreds of nodes in like a single unit, because like the problem becomes it's decomposed spatially, and so you have, you know, communication that has to happen at each step, like between all the different compositions, like across the nodes. So that's why we went for Frontera. But I'll also say, you know, Summit is a beautiful machine. Also, we get amazing runtimes on using NAMD. And then there are also some other applications which, you know, we actually have some collaborations now where we're gonna, I guess, try to run on Larson, which is, I think, maybe the number 4, one of the machines also on like the top 10 of the fastest ones, that's at Lawrence Livermore. But because we need to take advantage of something, you know, sort of, there's one particular application that we're looking at, or collaborators are looking at, where they need sort of a really sort of big node, I guess, to try to get it all on there and then do something with AI. So the choice of Frontera is, we've chosen to run the NAMD code for these big biological systems is going to be one of the best. For us, it's sort of one of the best, if not the best, code for doing that, and it runs really fast on these machines, including Frontera.
Question, how for someone who took, you know, chemistry more than 50 years ago in high school, can you, in very simple language, compare and contrast what you're studying compared to what the folding at home people are studying? How are they similar and how are they different?
Okay, they're actually pretty similar. So the folding at home people, they're studying actually a lot of different things, because now all of a sudden they have like so much compute time. So, you know, initially it started
SARS-CoV-2.
Yeah, no. But even within SARS-CoV-2, they're doing like all sorts of different things. But their main focus is, if I can say they have a main focus now, because I think there's like 3 or 4 different like major problems they're trying to look at. The main one was on like drug screening. you know, and so they would take a conformation of the protein, and they're trying to, like a game of Tetris, figure out which small molecules or potential drugs could like fit inside a particular site, right? So to try to, for example, block that handshake that I was telling you about. They're doing a lot of that, and those type of jobs, you know, you can like spit them out really quickly. They're small, relatively like not so intensive jobs, and they're one little unit, and you can spit it out to like billions of cores and then like grab it all back, right? They also, though, I guess are doing, I think that there's some segment of the cores that they have access to where they're also doing things like molecular dynamic simulations. As far as I know, so one of the differences is our model is really good of the spike. You know, we have it's like fully glycosylated. We've been very careful. We have they don't have, as far as I know, they have not simulated yet with the glycans, which is going to be very important for this particular protein. As I've showed you, it's a totally different structure without it. Those glycans are going to change the dynamics of the system, change the exposure of particular sites, et cetera. So it's important that they're there. They also don't have the stalk, and they don't have the membrane. So they're working on smaller sort of truncated systems or smaller other parts of the virus. So that's one of the major differences.
So maybe a corollary to that, you know, in our community, that we have some amount of you know, compute power, that, you know, one of the first reactions that people had was, you know, can we basically provide this for these types of computational activities? And, you know, folding at home has a certain visibility, so there was this sort of tendency that because it's set up in a trivial way, you know, at home, client, download a client and run it and just furnish the compute power, you know, there was kind of a rush in that direction. But yeah, what other, again, lacking a sort of, you know, high level community structure, the question becomes then, in what other places could, you know, we provide compute power that actually is potentially impactful?
I don't know, because to be honest, it's not so the way I see it, I'm sorry. I don't know if somebody wanted to say something.
I shouldn't interfere with your answer. Go ahead.
Oh, I was just going to say, to be honest, right now, before SARS-CoV-2, compute was a bottleneck. You know, it was harder to get the compute. We didn't have enough. Right now, everybody who has a processor in the world is basically like, use it for SARS-CoV-2. So that bottleneck of computing, it is not there. Right now, it's how are we going to use this computing to do something that's going to have impact? You know, unfortunately there's a lot of stuff I'll say going on that I don't think is probably going to have impact. You know, it's using up the time, and that's good, I guess. I don't know if it's good or bad, but it's happening. You know, and then so computing's not really a bottleneck. So off the top of my head, I don't really know. There might be particular novel application types, maybe something, you know, with machine learning where they need a particular architecture or something sort of very special. But
so,
I don't know, that's my take on it.
So that was something we started to perceive, was that potentially this was not the most useful contribution. So, you know, in the vein of having, you know, some number of extra, let's say, research software engineers, right, that are maybe not domain experts, but are, you know, software experts, and that, you know, maybe aren't incentivized in the same way in terms of, you know, the impact they need to have, that they contribute in this, you know, software sense. Where could they be impactful?
I think there could be in working with the MOLSI team to help, because you know, if we did have that, I mean, in some ideal case, you could imagine that all of a sudden now there would be like a small legion of people who could, in relatively short order, sort of help mature our community methods in terms of interoperability and this kind of thing, and also workflows, you know, because this is the other thing, like our work, because of the way that we do it, I mean, reproducibility, let's be honest, is an issue like in many areas of science, but like with molecular modeling, it's definitely been an issue for a really long time in molecular simulation. And that's in part because of the culture of the field where everybody kind of like holds everything in and doesn't share. And then when we do benchmarks, but it's on different data sets and it's just like, it's the Wild West. So, you know, perhaps then we can, you know, use this is what I had mentioned too, as an opportunity to get our field a significant movement forward in trying to, you know, get our methods to the point where they can be interoperable, where we can have, you know, sort of a workflow that is more automated, you know, that can be applied to different, you know, the same, so that we can actually like test the methods against, you know, multiple different data sets in a way that is like rigorous and like really tunable. That's where I think the development of that sort of infrastructure, which requires a lot of different, it's a lot of work and a lot of piecework. That's something I think could be great. So that's where I think, like, you know, working possibly together with MOLSI, if there are people who, you know, really want to do something and really want to help, that would be, I think, one great angle where maybe, you know, just there could be, you know, just some training sessions to get sort of a broad overview, and then the work could be potentially sort of, you know, divvied out or something. But yeah, I don't know.
So open it up to questions from anybody else also. Well, I have another question, but does anyone else have?
Well, someone said, yeah, that there is a meeting with BioExcel people to talk about CWL, that's that common workflow language probably, and have a sort of workshop. Yeah, that's fantastic. BioExcel, you know, so MOLSI and BioExcel kind of, I think, are a little bit similar, but, you know, BioExcel is overseas and the MOLSI's here, you know, and so this is really good. And I think that's a really good suggestion. And ideally, you know, we do this in sort of a global way. So thank you to Marcus Elsing for mentioning that.
Yeah, sorry, I really need to leave. That was very super interesting. I mean, that just happened today. I didn't make it to that meeting, but some of us were actually there, and we do have existing connections to what they're doing in BioExcel, and maybe something can develop out of this one.
Yeah, that'd be great.
Okay, so Rommie, this was great. Thank you very much for your time.
You're welcome.
We really appreciate it. Some of us may follow up afterwards. Again, looking at places where we can maybe marshal some effort to contribute. Maybe we may contact you. Thank you again.
That'd be great.
This is super interesting. Can I ask one or two more science questions?
Sure.
So when the LCLS was built at SLAC, it was designed to study the proteomics, the folding.
Yeah.
And all the data you talked about was static data, if I understand correctly. Is there any way to validate your calculations against the types of measurements that are made at SLAC or with similar facilities?
Yeah, I mean, that's definitely getting better to that. The thing with SLAC, yeah, so yeah, the data can, there's some, you know, that's at like sort of a pretty fine-grained scale of time.
Right, that's a small local scale.
Yes, but yes, absolutely, yes. You know, I think there it's just, you know, a matter of what applications are getting, you know, put onto those particular beams, lines, and then,
well, I'm just wondering, how do you validate your calculations generically? And what you said is, well, you know, someone has to go in the wet lab and actually make the modifications and see if they do what we predict.
Yeah, I mean, well, that's you know,
that's a macroscopic measurement.
Yes.
Where the stuff being done with these X-ray laser beams is really microscopic level, and that would be another. I'm just wondering whether you use that type of information in your science.
People do. I have not. I have personally not, but of course people do.
Okay, people do. That's what I'm wondering.
Yeah.
And then just about, say, the SARS-CoV-2 or influenza, whichever one you've studied. I guess you've probably studied the influenza more, but you've got these spike proteins here.
Yeah.
There are many of them on the surface.
Mm-hmm.
Are all the spike proteins on one virus particle, whatever you call it, are they all essentially the same, equivalent chemically, or are the variations from one spike to the next? Does the folding, does the chemistry depend on whether there's one physically really close to it, or if it's more isolated, because some of them are closer to others? In the picture behind you, for instance?
Oh, yeah, sure.
How much
Okay.
difference does that environment make?
Okay, so because there's two different questions here. So chemically, you know, at the level of like residues, they're very, very similar, very similar. But the issue is that then sort of more structurally and biologically, yeah, the local environment, you know, in some sense, you know, all these proteins, they basically do adopt the same fold. They look similar, but they're going to have different configurations, you know, like if you look at all the spikes on the virus, not all of them are going to be in like one receptor up conformation. There's like an ensemble. One, you know, some could be down, one could be up. One of the spike trimers could have all three up, you know, so there's some more at sort of a biological level, they start to look a little bit, they start to look different. And yeah, the environment, like in the virus behind me, which is influenza, it can matter. It can matter, of course, because, you know, you have actual like neighboring interactions. And so again, it kind of depends on like the scale of the motion and the phenomena that you're looking at. At the level of chemistry, it's very local. It's so local that it's hard to say that the scale at the whole virus, you know, really matters. But there's a lot of questions more on creeping towards what we think of as biology. Yes, where the environment does matter, and especially with like molecular recognition and so forth, if that makes sense.
It does. So fantastically interesting talk. I'm sorry that it is so urgent that you're studying this stuff today, but intellectually, it was a really, really interesting talk.
Good, thanks. Oh, you're welcome, thanks. All right.
Okay, well, thank you for your time and your effort on our behalf, and we'll be in touch.
Yeah, thank you. Take care.
Bye, everybody. Thanks.
Bye.
Thanks.